Good morning everyone. I am CH Vidya, Assistant Professor of Biochemistry, Government City College, Hyderabad. And today I am here to deliver a presentation on the topic Insulin, Mechanism of Action and Physiological Significance. The contents I am going to deal in this presentation are as follows. First I am going to deal with history, then biosynthesis and structure of insulin, physiological role of insulin, mechanism of action and deficiency disorder. So first I would like to throw light upon the significant contributors in the field of insulin research. So let us start with Paul Langerhans. Paul Langerhans was a medical student in Berlin in 1869. He was studying the structure of pancreas under the microscope when he noticed tissue clumps scattered throughout the pancreas. He named them as islets of Langerhans. Next, coming to Oskar Minkowski. In 1889, Polish-German physician Oskar Minkowski, in collaboration with Mering, removed the pancreas from healthy dog to test its assumed role in digestion. On testing the urine, they found sugar in the dog's urine, establishing for the first time a relationship between pancreas and diabetes. Then in 1916, a Romanian professor, Nicol Polesc, was the first to isolate the insulin. However, because of his anti-Semitic views, he has been erased from the history of insulin research. Then in 1920, Canadian Frederick Banting, reading one of the Minkowski's papers, assumed that the pancreas' internal secretions, which it was supposed regulates the sugar in the bloodstream, might hold the key to the treatment of diabetes. Then in 1921, Banting traveled to Toronto and with the help of J.J.R. MacLeod, the professor of physiology at the University of Toronto, and Charles Best, the lab assistant, worked on his idea. Then first insulin injection was made in the year 1922. And in 1923, the Nobel Prize Committee awarded Banting MacLeod the Nobel Prize in Physiology for the discovery of insulin. Banting shared his prize with Best and MacLeod shared his with James Collip, since they too were the contributors in insulin discovery. So these are the people who were involved. They are MacLeod and Banting. Then it is Charles Best and James Collip. Other significant contributors are Frederick Sanger. In 1958, British molecular biologist Frederick Sanger determined the primary structure of insulin. It was the first protein to have its sequence to be determined. He was awarded the Nobel Prize in Chemistry for his research work. Then it's Dorothy Crawford Hodgkin. In 1969, Dorothy determined the spatial conformation of the insulin molecule, that is the so-called tertiary structure, for which she was awarded Nobel Prize in Chemistry in 1964 for the development of crystallography. Then in 1977, Rosalind received the Nobel Prize in Medicine for the development of radioimmunoassay for insulin. Then Herbert Boyer was the scientist who first succeeded in making a synthetic human insulin through gene engineering in laboratory in 1977. So next we'll study about the structure and biosynthesis of insulin. Now insulin is produced by the pancreas, which has two important functions. One is producing hormones and the other is producing pancreatic digestive enzymes. Now here we can say that pancreas is a mixed gland. That is, the endocrine portion of the pancreas produces the hormones, whereas the exocrine portion of the pancreas produces the digestive enzymes. So you can see a cross section of the islets of Langerhans regions of the pancreas. The adult pancreas is made up of a collection of cells called islets of Langerhans, and there are around 3 million islets. There are 5 major cell types in the islets of Langerhans. They are alpha cells, beta cells, gamma cells, delta cells and epsilon cells. Alpha cells secrete glucagon, beta cells secrete insulin, gamma cells secrete pancreatic polypeptide, delta cells somatostatin, and last is the epsilon cells which secrete the hormone ghrelin. So as I already told you, insulin is produced by the beta cells in the islets of Langerhans, which release insulin in two phases. The first phase release is triggered in response to rising or increased blood glucose levels. The second phase is a sustained slow release of newly formed vesicles triggered independently of sugar. So beta cells in the islets of Langerhans take up as much as 60 to 80% of all the cells present in the islets of Langerhans. So formation of insulin. So coming to the formation of insulin, the genes on the chromosome 11 coding for insulin are first transcribed to mRNA in the nucleus, that is the messenger RNA. The messenger RNA which is present in the nucleus, it moves to the cytoplasm, and in the cytoplasm the mRNA is translated by the polysomes attached to the endoplasmic reticulum, resulting in the synthesis of pre-proinsulin, which contains 109 amino acids, which later enters into the lumen of the endoplasmic reticulum. Now inside the lumen of the endoplasmic reticulum, the pre-proinsulin is converted into proinsulin. Now if you see, the first structure is of pre-proinsulin. The pre-proinsulin consists of a signal peptide, a beta chain, the C chain and the A chain. A chain is alpha chain, B is beta chain, and C chain is the connecting peptide which connects the alpha chain and the beta chain. Inside the lumen, the pre-proinsulin is hydrolyzed by a peptidase enzyme which cleaves the signal peptide, and later two disulfide bridges are formed between the A chain and the B chain, resulting in the formation of proinsulin. So proinsulin consists of the B chain, the A chain, connecting C peptide, and two disulfide bridges between the A chain and the B chain. Then later the proinsulin is translocated to the Golgi apparatus, and inside the Golgi apparatus the proinsulin is again hydrolyzed by certain enzymes such as peptidases, and this peptidase will cleave off the C chain, that is the connecting peptide, and later it is again acted upon by another enzyme called as carboxypeptidase, which will remove the arginine residues present at the terminal regions of the alpha chain and the beta chain, and the resultant product is insulin. So you can see at the last there is insulin structure. Insulin is made up of totally two chains now, connecting with two disulfide bridges. So proinsulin is made up of 86 amino acid protein, and it has two chains linked together by connecting peptide. Insulin, it is made up of 51 amino acids, and it has two chains. One is the alpha chain and the other is the beta chain. They are connected by two disulfide bridges. Now if you see the insulin composition, as I told you earlier, it is made up of amino acids. You can see here the A chain consists of 21 amino acids, and the B chain, that is the beta chain, consists of 30 amino acids, together 51 amino acids. Now the two disulfide bridges you can observe here, and the molecules, the amino acids involved in this disulfide bridge formation are cysteine. Now we can see in the picture, the cysteine at the 7th position of the alpha chain is involved in the disulfide bridge formation, with the cysteine at the 7th position in the beta chain, and the other disulfide bridge is formed between the cysteine at the 20th position of the alpha chain and with the cysteine at 19th position of the beta chain. There is also an intra-disulfide bridge which is formed in the alpha chain between the cysteine at the 7th position and the cysteine at the 11th position. So insulin is a peptide hormone with a molecular weight of 5,808 Daltons. Insulin is produced and stored in the body as a hexamer. Hexamer is a unit of 6 molecules. In case of insulin, it is a unit of 6 insulin molecules, while the active form is the monomer. So storage form of insulin is a hexamer, whereas the active form is the monomer. Now let me discuss about the glucose-induced insulin secretion. Now insulin is secreted from the beta cells. Now the main trigger for the insulin secretion from the beta cells is the glucose presence, increased glucose levels in the plasma. So when there is increased glucose levels in the plasma, the glucose is driven into the beta cells through certain transporters called as glucose transporters. And the glucose transporter helps in the entry of the glucose into the beta cell. Now once the glucose enters into the beta cell, it undergoes metabolism. So the first enzyme which acts on the glucose is glucokinase. We come across glucokinase in the glycolysis pathway, whereon later several metabolic reactions take place, and at the end ATP is generated. Now this ATP generated inside the beta cell, there is an increase in the ATP levels. When there is an increase in the ATP levels, this ATP will bind to the ATP gated potassium channels. Binding of ATP to the ATP gated potassium channels will prevent the exit of potassium ions from inside of the cell to the extracellular environment. So when there is a prevention of the exit of potassium ions, obviously the potassium levels inside the cell would increase, and there is a disturbance in the electrical or the ionic charge between inside and the extracellular environment. This disturbance creates, this disturbance in the membrane potential will cause depolarization of the cell membrane. Now when the cell membrane gets depolarized, the voltage gated calcium channels will get stimulated, and calcium from outside of the cell will enter into the cell, and the calcium ions inside the cells would increase. Now the increase in calcium ions will help in the translocation of the insulin vesicles to the cell membrane. As I already told you, insulin is stored in the form of vesicles inside the beta cells. So whenever there is a trigger, these insulin stored vesicles will be translocated to the cell membrane, and they will be released by a process called as exocytosis. And in this way, insulin is released from the beta cells. Next is major factors regulating the insulin secretion. So previous slide we can see that there is an increase in glucose level, we have the insulin secretion. So here the high blood glucose levels will stimulate the pancreas, and when pancreas is stimulated, the beta cells would release the insulin, and the insulin will majorly act on different organs. Here you can see the picture of the fat cells, and glucose uptake takes place in these cells, and a normal glucose levels is achieved. In the same way, when there is low blood glucose levels, again the pancreas is stimulated, but the cells which are stimulated here are alpha cells. So the alpha cells get stimulated, glucagon hormone is released, glucagon acts on liver and results in the release of glucose into the blood, and thereby normal glucose levels are achieved. So you can see there is a direct feedback effect of plasma glucose on the beta cells of the pancreas. Next we will discuss about the physiological role. So here I am going to discuss the physiological role, that is the effect of insulin broadly on three main organs, that is liver, muscle and adipose tissue. So first I would like to tell you about the effects of insulin on the liver. In liver, it stimulates glucose uptake, it stimulates glycogenesis. Glycogenesis is nothing but formation of glycogen from glucose. Insulin stimulates protein synthesis, and it also stimulates lipid synthesis, and it inhibits certain biological pathways as well. So the insulin inhibits glycogenolysis, which is breakdown of glycogen for the formation of glucose. It inhibits ketogenesis, that is formation of ketone bodies from acetyl-CoA, and it inhibits gluconeogenesis, that is formation of glucose from non-carbohydrate sources. Effect of insulin on the skeletal muscle. In the skeletal muscle, the insulin stimulates the transport of free amino acids into the muscle. It stimulates protein synthesis and reduces release of amino acids from the muscle. It also stimulates glycogen synthesis. Then effect of insulin on fat cells. In fat cells, it reduces the rate of release of free fatty acids from adipose tissue. It stimulates de novo synthesis of fatty acids and also conversion of fatty acids to triglycerides in liver. Triglycerides is the storage form of fatty acids. Effects of insulin. So here you can see the effect of insulin, which is time based. So there is a rapid effect of insulin which occurs in seconds, and the actions are increased transport of glucose, amino acids and potassium ions into insulin sensitive cells. The intermediate effects which happen within minutes are stimulation of protein synthesis, inhibition of protein degradation, activation of glycogen synthase, increased glycogenesis, inhibition of phosphorylase and gluconeogenic enzymes. Then delayed actions, which means it takes few hours. They are increase in mRNAs for lipogenic and other enzymes. Now let's see insulin control. When there is an increased serum glucose levels, the beta cells of islets of pancreas will get stimulated. This causes release of insulin. Insulin will act on insulin sensitive organs. Here I am showing you liver and muscle. This causes glucose uptake, thereby causing normalization of the glucose levels. When there is a decrease in the serum glucose level, it acts as a negative regulator of insulin release. That is, when serum glucose levels decrease, insulin release was also decreased. Other activators of insulin release from the beta pancreatic cells are serum amino acids and gut hormones. In the same way, the negative regulators for insulin release are glucagon, somatostatin and other alpha adrenergic components. The next we will discuss about the mechanism of action of insulin. Based on the chemical nature, insulin belongs to the class of protein hormones. We have seen in the previous slides, insulin is made up of amino acids. So it comes under the class of protein hormones. Based on the mechanism of action, insulin belongs to group 2 hormones. Now here, basically based on the mechanism of action, we have two kinds of classes, group 1 hormones and group 2 hormones. Group 1 hormones are steroidogenic in nature. They are lipophilic, they can cross the cell membrane and they bind to the intracellular receptors to bring about the course of action. Whereas group 2 hormones such as insulin, which are proteinaceous in nature, they are lipophobic. So they cannot cross the cell membrane, so they have to bind to the cell surface receptors itself. So group 2 hormones bind to the cell surface receptors and stimulate the release of certain molecules, namely the secondary messengers, which in turn perform the biochemical functions. So this is a brief overview of how insulin functions. Insulin binds to the alpha subunit of the receptors. Autophosphorylation of beta subunit takes place. Changes in cytoplasmic proteins and enzymes will take place, which could be activation or inactivation, and at the end insulin exerts its action. So this is the receptor to which the insulin binds. Insulin receptors present in almost all cells. It is glycoproteinaceous in nature and it is a tetramer. It means it is made up of 4 polypeptide chains. The outside portion, that is the extracellular portion of the insulin receptor, is made up of 2 alpha subunits, whereas the other 2 subunits traverse the membrane and major portion of the beta subunit is present in the cytoplasmic side. Alpha subunit is the region to which the insulin hormone binds, and that is the alpha subunit has the hormone binding domain. Beta subunit has ATP binding and tyrosine kinase domain. So when insulin binds to the receptor, inside the cell there are 2 signaling pathways which can be stimulated. One is the MAP kinase signaling pathway and the other is the PI3K signaling pathway. MAP kinase means mitogen activated protein kinase signaling pathway. Activation of this pathway will lead to insulin targeted gene expression, whereas stimulation of PI3K signaling pathway, which is phosphoinositide 3 kinase signaling pathway, leads to synthesis of lipids, proteins and glycogen, and also cell survival, proliferation and another one is translocation of glucose transporters to the cell membrane for the uptake of glucose. And the glucose transporter which gets translocated in this process is GLUT4. So first I would like to discuss about the PI3K signaling pathway. Now before going into the pathway, I want to tell you about the downstream signaling molecules which we are going to encounter in the PI3K signaling pathway. The first one is IRS, that is insulin receptor substrate. The second one is PI3K, that is phosphatidyl inositide 3 kinase. Next is PIP2, phosphatidyl inositol 4,5-bisphosphate, PIP3, phosphatidyl inositol 3,4,5 triphosphate, then PDK, which is PIP3 dependent kinase, and AKT or PKB, which is protein kinase B. So here again we will discuss about the glucose transporters, that is glucose enters the cells by facilitated diffusion with the help of glucose transporters, that is GLUT1 to 7 are the different forms of glucose transporters. GLUT4 is the glucose transporter in the muscle and adipose tissue which is stimulated by the insulin. Now you can see here when insulin binds to the glucose, when insulin binds to the receptor which is present on the cell surface, that is insulin receptor, the beta subunit will have inbuilt tyrosine kinase activity, because of which it gets autophosphorylated, that is the tyrosine residues will get phosphorylated, and to this tyrosine residues which get phosphorylated, the IRS, that is insulin receptor substrate, will come and bind, and again the tyrosine kinase activity on the beta subunit will phosphorylate the IRS residues or the molecules. Now the phosphorylated IRS substrates will again phosphorylate further the other intermediates present. The one which gets activated here is the PI3 kinase. Now PI3 kinase will convert the PIP2 to PIP3, that is phosphatidyl inositide to 4,5-bisphosphate to phosphatidyl inositide 3,4,5 triphosphate. Now this PIP3 will further stimulate another enzyme, that is PDK, and PDK, once it gets stimulated, it will further phosphorylate a downstream molecule, that is protein kinase B. The protein kinase B will activate the glucose glucose transporters translocation from inside of the cell to the cell membrane, so that the glucose transporters will get situated there on the cell membrane and will help in the glucose uptake inside the cell. Another way in which your insulin will help in regulating your metabolisms is, let me tell you about how insulin is going to regulate the glycogen synthesis. Now up till the PKB, that is protein kinase B activation, is the same. Later downstream what happens is the protein kinase B will inactivate another enzyme called as glycogen synthase kinase. Now glycogen synthase kinase, when it gets phosphorylated, it becomes inactive. When glycogen synthase kinase becomes inactive, it cannot phosphorylate its downstream molecule, that is glycogen synthase. When glycogen synthase is not phosphorylated, that is, it is in its non-phosphorylated form, it is active in nature. And the active glycogen synthase will convert the glucose into glycogen, and in this way insulin will regulate the glycogen synthesis. And there is again transport of glucose into the intestine and kidneys is by secondary active transport with the help of sodium, that is sodium dependent glucose transporters. Next we will see about MAPK signaling pathway, that is mitogen activated protein kinase signaling pathway. The molecules which we are going to encounter in the MAP kinase signaling pathway are IRS, that is insulin receptor substrate, SHC, SRC homology and collagen, GRB2 growth factor receptor bound protein, SOS son of 7-less which encodes the guanine nucleotide exchange factor, And next is RAS protein. RAS protein has inbuilt GTPase activity, and it has been given the name RAS because it was first discovered in the rat sarcoma. The next is RAF kinase. RAF is rapidly accelerated fibrosarcoma. It was first discovered in the retrovirus, and it also has a kinase activity. It will phosphorylate serine and threonine residues on its substrate. So it comes under the class of serine threonine kinases. The next is MAPKK, that is mitogen activated protein kinase. Next is MAPK, mitogen activated protein kinase, and the last one is ELK, that is transcription activator. It has ETS domain. So this is the MAP kinase signaling pathway. So you can see the insulin comes and binds to the insulin receptor. It undergoes conformational change, that is the insulin receptor undergoes conformational change. The beta subunit undergoes autophosphorylation. Once the beta subunit undergoes autophosphorylation, the phosphoryl residues will, because of the presence of phosphoryl residues on the tyrosine molecules, the downstream molecules, that is IRS, will get stimulated. And the other molecule which gets stimulated in MAP kinase is SHC. So either IRS gets stimulated or SHC gets stimulated. Stimulation is nothing but the phosphorylation of these two molecules happens. So once they undergo phosphorylation, downstream there are other molecules that get stimulated. So the first one which gets stimulated or activated is GRB2. Now GRB2 will come when it gets activated, that comes and binds to another molecule, that is SOS, which has guanine nucleotide exchange factor. Now these two form a complex, that is GRB2 SOS complex. This complex will convert RAS GDP into RAS GTP. Now here I want to say the RAS protein, when it is bound to GDP, it is inactive in nature. But when the GDP is exchanged with GTP, that is RAS GTP, that is active in nature. So here GRB2 SOS complex will exchange the GDP for GTP for the formation of RAS GTP, that is active form. Now the RAS GTP which is active will further activate another downstream molecule, that is RAF kinase. RAF kinase gets phosphorylated. The phosphorylated RAF kinase further phosphorylates MAPKK. It's another kinase. The phosphorylated kinase further phosphorylates MAPK, that is mitogen activated protein kinase. The phosphorylated mitogen activated protein kinase at the end phosphorylates ELK1, which is a transcription activator. Now the phosphorylated ELK1 enters into the nucleus, binds to the insulin target genes, and causes the transcription of the insulin target genes. So in this way, insulin through the MAP kinase signaling pathway is causing increase in gene expression of its target genes. So now I am going to tell you about the deficiency of insulin. In the previous slides, when I was telling you about the physiological role of insulin, I was discussing about the effect of insulin on three main organs, that is muscle, liver and adipocyte. So here I am going to tell you the effect of deficiency of insulin on three organs. That is, first let me say about muscle. Muscle, it can't use glucose and amino acids, so it goes into a stage of starvation. Potassium and phosphate is not normally transported when there is a deficiency of insulin. The next on liver, more glucose is made in the liver. Then effect of deficiency of insulin in adipocyte, lipolysis goes unchecked, that is the triglycerides breakdown takes place, free fatty acids are released into the bloodstream. These free fatty acids undergo oxidation for the release of ATP. So the insulin disturbances are seen in many pathological conditions. They are diabetes mellitus, PCOS, polycystic ovarian syndrome, metabolic syndrome and insulinoma. Out of all this, I want to discuss about diabetes mellitus. We are aware of the fact that deficiency of insulin or any disturbances in insulin leads to diabetes mellitus. What is diabetes mellitus? Diabetes mellitus is a group of metabolic diseases in which a person has high blood sugar either because the blood does not produce enough insulin or because the cells do not respond to the insulin that is produced in the body. Classification of diabetes mellitus. Diabetes mellitus is classified into 4 types. Type 1 diabetes mellitus, which is autoimmune in nature, idiopathic in nature. Type 2 diabetes mellitus ranges from relative insulin deficiency to disorders of insulin secretion and insulin resistance. Other specific types of diabetes mellitus are genetic defects in beta cell function, genetic defects in insulin action, diseases of the exocrine pancreas such as pancreatitis, pancreas trauma, cystic fibrosis, endocrinopathies such as acromegaly, Cushing's syndrome, hyperthyroidism, infections, or sometimes pharmacologically or chemical induction also takes place. Then gestational diabetes mellitus, it occurs mostly in the women during gestation. So here you can see type 1 diabetes mellitus, genetic susceptibility, environmental factors, viral infections will lead to the production of autoantibodies against the beta cells, causing beta cell destruction, insulin deficiency, and in the end type 1 diabetes. If you see type 2 diabetes, beta cell defect, secretory defect, excess food, obesity, sedentary lifestyle, and sometimes low birth weight will lead to relative insulin deficiency, insulin resistance in target cells, beta cell exhaustion, and type 2 diabetes. This is a comparative picture of how a healthy individual will be and type 1, type 2 individuals. Now, if in a healthy individual the insulin levels are normal, so the glucose will normally enter into the cell and thereon insulin will carry out its function normally. Whereas in type 1 diabetic patients, pancreas fail to produce insulin, so glucose cannot enter inside the cell. Whereas in type 2 diabetes, insulin may be produced normally, but the cells are unable to respond to the insulin properly. Now, differences between type 1 and type 2 diabetes. Type 1 is insulin dependent diabetes mellitus. Type 2 is non-insulin dependent diabetes mellitus. Type 1 occurs during childhood. Type 2 occurs at the onset of age after 35. The pattern of onset is abrupt in case of type 1 and it is slow, that occurs gradually in case of type 2. Genetic predisposition is moderate in type 1, very strong in type 2. Nutritional state, undernourished in type 1, obese in type 2. Biochemical defects are autoimmune destruction of beta cells in case of type 1. Insulin resistance combined with inability of beta cells to produce appropriate amount of insulin is in type 2. Plasma insulins are almost low to absent in type 1, whereas in type 2 it is high in early stage, but as the disease progresses it becomes low. The frequency of ketosis is very common in type 1, whereas it is very rare in type 2. The treatment here is insulin is always needed in case of type 1 diabetic patients, whereas in type 2 diabetic patients, diet, exercise and oral hypoglycemic drugs can be given as treatment. And next, these are the levels which we can observe. In a normal individual, the plasma glucose levels in the fasting stages around 70 to 100 mg per deciliter. If it is increasing, that is 100 to 126, it shows that the person is having impaired fasting glucose. Whereas if it is more than 126, he is suffering from diabetes. If we see the postprandial, if it is less than 140, the person is normal. If it is 140 to 200, the person is having impaired glucose tolerance. If it is greater than 200 mg per deciliter, then the person is diabetic. So this is the picture of what are the effects a person is suffering when he is having diabetes, the symptoms and other complications involved when a person is having diabetes. So the fundamental defects are majorly there is a reduced entry of glucose into the cells, increased liberation of glucose into the liver, from the liver into the circulation, and there are various signs and symptoms in diabetes which majorly occur due to disturbances in carbohydrate, protein and lipid metabolism. Now let us see the consequences of disturbed carbohydrate metabolism. The major symptoms which we can see here are polyuria, polydipsia and polyphagia. The renal threshold value For glucose, for the kidney, that is the renal threshold value for glucose is around 180 mg per deciliter. When there is an increase in the plasma glucose levels, that is more than 180 mg per deciliter, the glucose starts appearing in the urine, and that condition is called as renal glycosouria. So when glucose comes into the urine, it will bring along with it water, and that condition is osmotic diuresis. So glycosouria along with osmotic diuresis causes polyuria, that is increased frequency of urination. When a person is having polyuria, obviously there is a water loss which leads to dehydration. Now this dehydration will cause increased thirst in the person, and that condition is termed as polydipsia. So the quantity of glucose lost in urine is enormous, and thus to maintain energy balance, the patient takes in large quantities of food, and this leads to a condition called as polyphagia, which is excessive eating. Now consequences of lipid metabolism. So when insulin levels are low, the hormone sensitive lipase in the adipose tissue gets stimulated, fatty acid get released, the free fatty acids get released due to the process of lipolysis. The free fatty acids undergo oxidation and acetyl-CoA is released. Acetyl-CoA has to enter into the tricarboxylic acid cycle or Krebs cycle. But since there is an overload of TCA cycle, it gets saturated, and the excess acetyl-CoA are involved in the ketogenesis pathway, that is ketone bodies. So the ketone bodies which are produced in our body are acetoacetate, acetone, and beta hydroxybutyrate. Ketone bodies are relatively strong acids, and their increase in the body lowers the pH of the blood. So when the pH of the blood gets decreased, the condition is called as acidemia, which impairs the ability of binding of hemoglobin to the oxygen. So here is the overall picture of what happens when there is a decrease in the insulin levels on the lipid metabolism. So I have told you there is an increase in the ketone bodies. This ketone bodies again escape into the urine, leading to ketonuria condition, and the decrease in the pH of the blood takes place, called as metabolic acidosis, and this may lead to Kussmaul breathing, a very deep and burdened breathing, fruity odor, and sometimes if left untreated may cause coma and eventually death. Consequences of disturbed protein metabolism: increased protein breakdown will cause muscle wasting, increased protein synthesis, increased plasma amino acids and nitrogen loss in urine, leading to negative nitrogen balance and protein depletion. And protein depletion is associated with poor resistance to infection. These are the consequences of disturbed protein metabolism. Next is consequences of disturbed cholesterol metabolism. In diabetes, the cholesterol level is usually elevated, leading to atherosclerotic vascular diseases. Now this is due to a rise in the plasma concentration of VLDL and LDL, that is very low density lipoproteins and low density lipoproteins, which may be due to increased production of these lipoproteins by the liver or decreased removal from the circulation. So you can see the picture over there, normal coronary artery and atherosclerotic artery. The plaques are formed, they get situated in the blood vessel, and ultimately a clot is formed there. Next is the effect of diabetes in case of wound healing and gangrene. Here you can see there is a circulatory insufficiency due to atherosclerosis. So when atherosclerosis is taking place, a blood clot is being situated there, a plaque is being situated there, that will disrupt the flow of the blood in that area. And there is neuropathy condition, there is protein depletion which causes poor resistance to infections. So when there is protein depletion, the resistance of the body towards infections gets decreased. That leads to, the poor wound healing. And advanced glycosylation end products cause a decrease in leukocyte response to infection. Advanced glycosylation end products are formed when glucose binds non-enzymatically to certain proteins, and that to amino groups of the proteins. And these advanced glycosylation end products cause a decrease in the leukocyte response to infection. In this way, diabetes will lead to all these conditions, and these conditions will eventually lead to poor wound healing, and if left untreated may cause gangrene conditions. So let me summarize the whole presentation. So insulin is produced by the beta cells in the islets of Langerhans. Insulin is a peptide hormone. It is made up of 51 amino acids comprising of two chains, the alpha chain, which is made up of 21 amino acids, and the beta chain, which is made up of 30 amino acids, linked by two disulfide bridges. Before the insulin formation, I've told you already, there are other precursor molecules, that is pre-proinsulin and proinsulin. Pre-proinsulin is made up of 109 amino acids, whereas proinsulin is made up of 86 amino acids. The pre-proinsulin is converted into proinsulin in the lumen of the endoplasmic reticulum by the enzymes peptidase, wherein the signal peptide will be removed and disulfide bridges will be formed between the alpha chain and the beta chain, resulting in the formation of proinsulin. The proinsulin is converted into insulin in the lumen or in the Golgi apparatus, wherein the proinsulin having the C peptide, that is connecting peptide, is released, and the two chains, that is alpha chain and beta chain, connected by the disulfide bridges, that is the whole insulin molecule, is produced. The next is insulin is produced and stored in the body as hexamer, a unit of 6 insulin molecules, while the active forms is the monomer. In the previous slides we have seen that the insulin is stored in the vesicles, and I have already shown you the figure of how glucose-induced insulin release takes place and how the vesicles which contain the insulin are translocated to the cell membrane region, and insulin is released as monomers for its course of action to be carried out. The next is insulin is considered as an anabolic hormone. Now we can see in the previous slides that almost anabolic reactions or metabolisms were triggered by insulin. So insulin is majorly considered as an anabolic hormone. It regulates the metabolism of carbohydrates, fats and proteins by promoting the absorption of carbohydrates, especially glucose, from the blood into the liver, fat and skeletal muscles. Thereby so if you see here, the carbohydrate metabolic pathways which get stimulated by insulin are glycogenesis, and inhibited are glycogenolysis and gluconeogenesis. Whereas in case of lipid metabolism, we can see that lipogenesis is stimulated and lipolysis is inhibited. The reverse happens when there is a deficiency of insulin state. Insulin stimulates glucose uptake, glycogenesis, that is formation of glycogen from glucose, protein synthesis and lipid synthesis. Lipid synthesis is nothing but formation of your storage form of lipid, that is triglycerides, from the free fatty acids. Then insulin inhibits glycogenolysis, ketogenesis and gluconeogenesis. Insulin belongs to group 2 hormones. It brings about its biological function inside the target cell through two signaling pathways. One is PI3K signaling pathway and the other is the MAP kinase signaling pathway. Through PI3K signaling pathway, it is involved in the synthesis of lipids, proteins and glycogen. GLUT4 translocation to the plasma membrane for glucose uptake. Through MAPK pathway, insulin targeted gene expression takes place. The last one is deficiency of insulin leads to diabetes mellitus, which is characterized by hyperglycemia, glycosuria, polydipsia, polyuria, polyphagia, ketosis, acidosis, coma, eventually death if left untreated. Hyperglycemia is increased glucose condition, glycosouria is presence of glucose in the urine, polydipsia is increased thirst, polyuria is increased frequency of urination, polyphagia is increased hunger, ketosis is production of ketone bodies, acidosis is a condition where increased ketone bodies production will lower the pH in the blood.